Okay, so I'm going to talk to you about the history of microbiology. So, bacteria date back to 3.5 billion years ago. I'm probably not going to quiz you over that. I just think it's interesting that they're that old. And then eukaryotes, remember that's things that have a nuclei. So if you don't remember that, write that down. They have a nuclei. Appeared about 1.5 billion years ago. So if you think about it, bacteria, the prokaryotes, were here 2 billion years before eukaryotes appeared, right? That's crazy. The father of microbiology is supposedly that handsome dude on the right there. He built his own microscope. That is Anton von Leeuwenhoek. Microscope was pretty crude, but for its time, pretty awesome, really. And then Louis Pasteur and Robert Koch in 1861, they're considered microbiologists because they proved biogenesis, that living organisms come from other living organisms. And, before this, they kind of thought that if you had tuberculosis, you were a witch or whatever, and it was just spontaneously in the air. And they said, no, you have to have tuberculosis bacteria to then get tuberculosis, and then tuberculosis bacteria makes other TB bacteria, etc. Kind of a crazy concept. The people that disagreed with them, they were abiogenicists, people that said you don't need life to get other life. Okay, a little bit about the kingdom system that I think you read in the book. So there was kind of an old kingdom system back when I went to school. Basically 5 kingdoms. Bacteria were in the Monera kingdom, and then you had protists, little pond swimmies, etc., plants, fungi, and animals. And then this guy on the slide here, Carl Woese, came up with a better way to do it. He said that there should be something bigger than kingdoms. So he came up with a domain system. And I say new, but this really isn't that new. Some teachers still don't teach it, which shame on them. And that reflects a greater understanding of evolution and molecular evidence, again, proposed by Carl Woese. So he said that basically you should have 2 different domains for bacteria. There's one that's basically called bacteria, and there's archaebacteria. Those are really ancient, old bacteria. And then eukaryotes. And then underneath the eukaryote domain is pretty much anything that has a nucleus. So the 3 big domains are 2 are actually bacteria, and then the other one is just for anything else with a nucleus. This slide kind of shows a little bit about the 3 domains. So domain bacteria, domain archaebacteria, and then all the other stuff on this slide would be in the domain eukarya, which basically is anything that's a eukaryote, right? I'm not going to read this slide to you, but I do think it's important to note kind of the difference between the archaebacteria and the bacteria. So the archaebacteria, you might even highlight this example, the halobacterium. So think about it, halo refers to salt. So, they live in water with high salt concentrations, and archae refers to ancient, old. So they're kind of odd ducks. Okay, they're really, really old. They've been around for a long time. And then the eubacteria, or just bacteria, their U means true, so they are the true bacteria. So you might highlight an example. I think you guys know, you've heard of staph, right? Like a staph infection. So Staphylococcus aureus, that's a true bacteria. It's not ancient compared to the other guys. The other guys also, the archaebacteria, would be found in hot springs, like out there in Yellowstone. They like that kind of environment. And then eukarya, of course, this frog is a eukaryote. Okay, a little bit about prokaryotic organisms. They have DNA. It's a double helix, but it's circular instead of linear. Okay, so our DNA, if you can picture it in your head, it's linear, right? Well, theirs is double helix, but it's in a circle. And then they also have these cool little things called plasmids. So this is on top of the DNA that they already have, as shown in the picture on the bottom left. It is an extra chromosomal piece of DNA. Okay, so it's extra, and it can code for antibiotic resistance. The cool thing about those plasmids, well, I guess not cool for us, is they can transfer them. So think about it. If I'm a bacteria and I have a plasmid that says I'm going to be resistant to an antibiotic, and then I give it to another bacteria, Now they have the gene to become resistant to antibiotics, and that's how the whole thing starts, pretty much. I gave you a picture of the pea-glow plasmid. We're going to work with that, hopefully, next semester, and this is just kind of a plasmid map. You'll learn more about that next semester. But on this, you see the AMP-R, the little AMP and then the superscript R. That is the gene that allows the bacteria to become resistant to ampicillin. Ampicillin is just an antibiotic. And we're going to genetically engineer the bacteria that get this plasmid to glow in the dark. So, good example of what we can do with these plasmids. So this is just a picture of a run-of-the-mill bacteria, right? They have some organelles like ribosomes because they have to crank out proteins. They do have DNA, although it's kind of circular in there. It doesn't look like it in this picture, but it is. It's all kind of bunched up and circular. They do have cell walls, most of them, and then some of them on top of that have a capsule. We'll talk about that. And then some of them have flagella, some of them have a pili. So there's lots of different forms, but this is just kind of a good run-of-the-mill one. Okay, they do have a few structures in common. So they do have plasma membranes in common with eukaryotes. That plasma membrane is selectively permeable or semi-permeable, right? We talked about that in biology. It doesn't let everything in. They do possess ribosomes. They have to make proteins just the same as you do. The eukaryotic ribosome is made of two subunits, a 60S and a 40S. Okay, and you can see that in the bottom right, the 60S and the 40S. There's actually two little pieces that compose a whole ribosome. And then they kind of click together when they are making a protein, basically. Prokaryotic ribosomes are smaller. They are made up of 50 and 30S. So the subunits themselves are a little bit smaller, and that makes the total ribosome just a little bit smaller. And then the S, this might be worth extra credit or something. The S stands for Svedberg. It's a Svedberg unit named after the guy who kind of discovered these little pieces of the ribosome and that they weren't just a simple solid dot. So prokaryotic organisms also have cell walls, like I said. Those cell walls are different than plant cell walls. They contain peptidoglycans, and that's kind of a fancy thing that's basically a sugar and an amino acid smushed together. So if you think about it, peptido refers to like the peptide bond in amino acids. So that's kind of where the amino acid comes in. And glycan refers to sugar. So they have peptidoglycans. And those little orange tic-tac looking things on the picture, that is the peptidoglycan layer. And if you notice, that is on top of the plasma membrane layer, which is below that with the blue blobs in there. So remember, the cell wall goes on top of the plasma membrane. There is, and I should probably push escape here, hold on, kind of an interesting difference between gram-positive and gram-negative, and we'll probably get into that. But gram-positive has different linkages between, this is like if we zoom in on the cell wall versus gram-negative. So that's one of the differences between gram-positive and gram-negative, is the different linkages in these little structures. Okay. All bacteria have a cell wall except Mycoplasma. Those are odd ducts. They cause tuberculosis, amongst other things. The cell walls can help differentiate them by a gram stain, which I hope to do with you. That separates them into gram-positive and gram-negative. So if we look over here, the gram-positive cell wall has cell wall, cell membrane, but the gram-negative cell wall is, more complex, I guess. So this is just another look at the gram-negative and gram-positive. Gram-negative also has these little weird lipopolysaccharides kind of on the top of it. And we'll get into this probably a little bit more. Another example here of and I wanted to show you this one. So this is a really, really thick peptidoglycan layer of the gram-positive. I think it looks like a huge, Hershey's chocolate bar or something. And then this is the same peptidoglycan layer in the gram-negative, pretty tiny. But then they also have another layer on top of that, so it's more complex. This one's really thick and kind of dense. This one's probably thinner and more complex. Okay, let's get into the gram-positive cell wall just a little bit. So like I said, it's thick and dense. It doesn't have another membrane covering on top of that, and it makes them susceptible to most antibiotics. So gram-positive bacteria would be like staph, strep, your strep throat bacteria. It doesn't necessarily, this is just a blanket statement. There are lots of gram-positive bacteria that would be resistant to antibiotics, but most gram-positive will be wiped out by an antibiotic. So you might write a couple of examples down. Streptococcus, staphylococcus, those are 2 gram-positive bacteria. And you know they're gram-positive by the way they accept this stain, and that's called the gram stain. And they also stain purple. And we'll talk about why they stain purple. Really has to do with kind of that thick peptidoglycan layer. It really holds the stain well. Okay, gram-negative. They are multi-layered, membranous. Like I said, they're a little bit more complex. They have that thinner peptidoglycan layer, that cookie-looking layer there, the chocolate bar. They contain on top of the peptidoglycan layer another layer, and that layer often has these weird-looking lipopolysaccharides, which are those little green and blue flag-looking things. And then they are often surrounded by another envelope, an outer envelope. So they are more complex. Examples of these guys would be E. coli, salmonella that you get from, raw chicken and whatnot. You can get food poisoning from either one of those. They stain more pink because they don't have that thick peptidoglycan layer. The purple stain, because you stain both of them purple, but the purple stain washes off of this one because it's not retained in that really thick peptidoglycan layer. This is just a little more about each one of them, but I kind of liked this little bottom thing, this thing down here. If it's got a lipopolysaccharide outer membrane, it's negative, and if it's positive, it's got a thick peptidoglycan. We don't talk about that, but that's kind of a little memory device for the gram-negative and gram-positive, I guess. Just another picture. I like giving lots of pictures so you can refer to them. And then we'll talk about the gram stain, but this on the far left here shows the gram-negative. That stain ends up washing off. So you start staining them both purple, and then, like I said, it washes off because it's not very thick. And then in the gram-positive bacteria, the purple stain really is held in that really thick peptidoglycan layer. That's an actual slide shot of a gram-positive bacteria, the purple. That's staph. And then E. coli is the more pink kind of sausage-looking ones. Many, but not all bacteria have a capsule. That is an outside layer beyond the cell wall made of polysaccharides and proteins, and it protects the bacteria from chemicals, bacteriophages. Those are viruses that infect bacteria. So if you don't know what a bacteriophage is. And then desiccation. So desiccation means drying out. But good word, I want you to know it, desiccation. So this extra layer on top of the cell wall that not all bacteria have is called a capsule, kind of protects them